All right, welcome to the video lecture for Chapter 5. Your textbook, your online textbook, calls this cell structure, but this is really all about membrane transport, moving things across the membrane, either into the cell or out of the cell. We'll start by reviewing membrane structure, and that'll help us to understand how these various modes of transport are possible. And then we'll look at a couple methods of passive transport where we don't use any energy. And then we'll look at the methods of active transport that allow us to expend energy to move material into or out of the cell. So first, let's a quick review of membranes, and we spent a fair amount of time on this already. Remember, membranes are a phospholipid bilayer with all kinds of proteins, some on the outside, some on the inside, some spanning through the membrane. Remember, the membrane is a fluid mosaic. A mosaic just means that it's composed of a lot of different things. If you think about when you were in the third grade, you made a mosaic of kidney beans and corn kernels and peas on a piece of paper, and you gave it to your grandmother, and she put it on the fridge, and it's been there ever since. But that's kind of what a mosaic is. It's a structure made from a variety of different things. The membrane is semi-permeable, meaning that some things get through and some things don't. And again, in animal cells, there's a significant amount of cholesterol in the tail region of the membrane to give it strength but still flexibility. The membrane defines the outer border of all cells, and if the cell has membrane-bound organelles, eukaryotic cells, then it defines the outer border of those organelles. It also helps to determine what can enter and exit the cell. Because of the properties of the cell membrane, some things aren't going to get through unless a protein moves them through. Other things can pass through easily. Because of the proteins that are in the membrane, it can respond to external signals and initiate a cellular response, and it can help to anchor to neighboring cells depending on whether there are junctions or a middle lamella or other things, or a cell wall, other things associated with the cell membrane. The key to membranes is understanding the dual polarity of phospholipids. And I know we've talked about this before, but it's not a bad idea to hit it again. Phospholipids have the two long-chain fatty acids, which are hydrophobic, that don't interact with water, and they have the hydrophilic head. Anytime you see lots of oxygens, okay, all those oxygens can hydrogen bond with water. And so this part of the molecule is hydrophilic and can hydrogen bond with water, and this part can't. And if we took a bunch of phospholipids and we put them in a glass of water and we shook them up, they would form these bubbles with a bilayer membrane with water on the inside and water on the outside. It's their chemical nature to form this structure, okay? And so it makes a really nice membrane. Anything that can interact with the polar head probably won't be able to interact with the nonpolar tail, and vice versa. Now, membrane transport are ways we can get things across the cell membranes. And it's important that we get things across the membrane because we need to bring nutrients into the cell, we need to get waste materials out of the cell, and if the cell is producing a product, like your beta pancreatic cells produce insulin, that they need to excrete from the cell into the bloodstream so that it can travel through the blood and help your cells to pull glucose out of the blood. If you can't produce this, you suffer from diabetes. And so if you're a cell that is going to secrete something, it's really important to have membrane transport, but all cells need membrane transport. This allows the composition of the cytoplasm to be different from what the cell is in. And there are two types of transport: passive, where you don't use any energy to move something across the membrane, and active, where you have to use energy from either an energy-rich molecule like ATP or by utilizing charge gradient. The first method we'll talk about is a passive method, and that's simple diffusion. Simple diffusion occurs when particles move from where there's a lot of them to where there aren't. And this is really common. You know, if you're making tea, and you take a tea bag and you put it in a cup of hot water and you just let it sit, you can see the tea slowly start to diffuse out of the tea bag and into the cup of water. And if you wait long enough, the tea will be an equal concentration everywhere in the water. And the same thing happens with particles that are really small and nonpolar, like diatomic oxygen and carbon dioxide, the gases. The small lipid hormones can also diffuse through. And what happens is they start as in a large concentration on one side of the membrane, and then they don't want to all be there. They're bouncing against each other and they're pushing each other. And as some of them start to move through, eventually you have equal concentrations on both sides of the membrane. And so now as one leaves from the bottom to go up, one from the top will leave to come down and maintain that equilibrium. And simple diffusion is going to continue until you reach equilibrium. When water diffuses, we call it osmosis. And of course, this was a big part, or is a big part of the diffusion lab activity that you may or may not have already done in the lab with your instructor. But when water is in solution, a solution is a homogeneous mixture of a solvent, whatever's in the greatest amount, and all the solutes, all the things dissolved in the water or in the solution. You could think of the air in the room as a solution. It's mostly nitrogen, 80% nitrogen, but it's got some oxygen and carbon dioxide and water vapor and dust particles and viruses and bacteria floating around. And when you look at the air, you really can't see all the different components. That's why it's homogeneous, same makeup throughout. Okay, but whatever's in the greatest amount is a solvent. In blood or in the cytoplasm or in the interstitial fluid between your cells, water is the solvent. And all the things dissolved in the water are solutes. Now, water can pass a semi-permeable membrane, but the solutes may not be able to. And so if we start here in this beaker, they've actually separated the two sides of the beaker with a semi-permeable membrane. One side, the pink is the water, the little round purple spheres are solutes. And one side has many more solutes than the other. Water wants to be at equilibrium. On this side, there's more water to solute than there is on this side. And so it wants to be equal. So water's going to move. It's going to move across this membrane until it's at equilibrium. Okay? Even though it's going to be pushing the water up higher on this side than it is here. This creates pressure. And so cells can use ions and pump ions to one side of the membrane or other to force water to move into or out of a cell or cellular structures. This is how your kidneys function. Tonicity is the ability of a solution to cause the cell to gain or lose water. And there are three different solutions that a cell could find itself in: an isotonic solution, a hypotonic solution, and a hypertonic solution. In an isotonic solution, the ratio of water to solute is equal on both sides of the membrane. So water flows freely in both directions, and the cell doesn't change. This is a red blood corpuscle, and they typically have this round, but with a sunken middle. It doesn't go clear through. It's not like a donut, but it's kind of a sunken depression there. And this is how it looks normally when it's doing a good job carrying oxygen in your bloodstream. If we put the cell in a hypotonic solution, where there's less solute outside the cell than in the cytoplasm, some water is still going to leave, but far more water is going to come in, and this cell is going to swell up, and it may not function as efficiently. If it really swells up without a rigid cell wall, it might burst. In a hypertonic solution, there's far more solute far more solute outside the cell than inside the cell. And so what happens is some water comes in, but most water is leaving the cell. This cell is going to shrink, and it's not going to be nearly as active as it usually should be. This is why you can't drink salt water. This is why if you have a diet that's heavy in pop or beer or salty beverages or sweet beverages, it's actually dehydrating water from your tissues and making you thirstier. Now, cells use tonicity to they want to control this, and we'll see this in the next slide. But again, in an isotonic cell, we've got an equal concentration of solute. So the cytosol has the same osmolarity or concentration of solute. Water flows equally in both directions. Cell doesn't change. When you need an IV, they're going to give you an IV at 0.9% sodium chloride, which matches the tonicity of your cytoplasm so that your cells don't gain or lose water. In a hypotonic situation, water is going to flood into the cell, and cause a cell to swell. In a hypertonic solution, the excess water is going to leave the cell. Animal cells function best in isotonic situation. Plant cells actually function best when it's hypotonic because they want that excess pressure in to fill their central vacuole and to create that turgor pressure on the inside of the plant cell. Again, cells very much need to balance the water balance between what's inside the cell and what's outside. This is called osmoregulation. And we do this on the whole body level. This is what our kidneys do, okay? But at the cellular level, there are single-celled organisms like this paramecium that have structures called contractile vacuoles. And see, what happens is the cytoplasm is hypertonic to the environment. Water wants to flood in. And the paramecium doesn't have a rigid cell wall, and it doesn't want that high internal pressure. So what it does is it collects excess water from its cytoplasm in the contractile vacuoles and pushes it out of the cell. So in this single-celled organism, they have organelles that function very much like our kidneys. Plant cells want to be in a hypotonic environment. And if they end up in a hypertonic environment or even an isotonic environment, they're going to get wilty. And you can see this plant has lost water and is starting to wilt. But as soon as we water it, the plant is now in a hypotonic situation, and the stems and leaves become turgid again, become full. Their central vacuoles are full of water. They're pushing that turgor pressure that helps keep the plant erect, which is important if you're reaching up to the light. Both plant and animal cells are going to suffer in a hypertonic environment. They're both going to lose water and shrivel. Another form of passive transport is facilitated diffusion. And it's very similar to simple diffusion. Both are passive, so they don't use any energy. Both follow the concentration gradient, moving things from high concentration to low concentration, and both move things across the membrane. The difference is that in facilitated diffusion, you need help from a protein to do this. You need either what's called a channel protein, which creates a pore to allow material to quickly pass through, or a carrier protein that's actually going to bind to something, and when it binds, change its shape to create a portal to move something through. And this is how cells move ions and small polar molecules into or out of the cell. Channel proteins, sometimes they're open all the time, other times they're gated, where the cell can close the gate to keep them from allowing whatever would rapidly come through. One of the best examples of channel proteins are aquaporins. Remember, water's a polar molecule. It's got a slightly positive hydrogens and slightly negative oxygen. And because of that, it doesn't pass through this hydrophobic tail region very well. It's very, very slow. So if the cell needs to speed up the flow of water into or out of the cell, it can open up the aquaporin, and now water can readily move through and get to equilibrium faster. Carrier proteins, again, remember, proteins have shape, they have structure, primary, secondary, tertiary, and quaternary, and this results in a three-dimensional shape. And when something can bind to the protein, it can alter the bonds that give it that three-dimensional shape. And then the protein changes its shape, and in the process of changing its shape, can create a portal from one side of the membrane to the other. And so in this type of facilitated diffusion, binding from high, again, we're in high going to low concentration, but when this solute particle binds, protein changes its shape, creates a portal. Once it releases it, now it reverts to its former shape because its internal bonding patterns can fix again. And this allows movement in either direction. It just depends on where the concentration gradient is, where whatever we're moving is in a high concentration. Different from passive transport is active transport. Active transport, we're going to spend energy. This is necessary when we're either transporting an ion or molecule against a concentration gradient, so from low concentration to high, we're pushing it to the high side, or against electrochemical gradient, meaning to push, in this example, positively charged hydrogen ions to where there are more positively charged hydrogen ions. They have the same charge, they're repelling each other, they don't want to be together, so it takes energy to push them that way. In terms of active transport, there's primary active transport where we're getting our energy from ATP, like you see up here on the proton pump in the diagram. And then secondary active transport where an electrochemical gradient provides the energy. Here you can see as these we've pushed the protons to this side of the membrane. They don't all want to be here, so they're going to diffuse back through. And when they diffuse through this protein, they take sucrose with them to the inside of the cell. In terms of active transport carrier proteins, these are integral proteins that are embedded through the membrane, and we call them pumps. And there are three types of pumps. A uniporter carries one thing at a time across the membrane in either direction, but one thing at a time. A symporter is going to carry two different things across the membrane at the same time, but in the same direction, either both coming in or both going out. An antiporter is going to carry two different molecules or ions, but it's carrying them in different directions. As one thing comes through to the inside of the cell, something else is going out of the cell. In examples of uniporter and symporter pumps, we'll go back to our diagram that we saw before. In the proton pump, hydrogen ions, this pump is acting as a uniport. And what's happening is hydrogen ions are binding, and then ATP comes along and binds and gives up a phosphate to the pump, and that adding the phosphate to the protein changes its shape, allows it to push those protons, those H pluses, from where there's very few of them out to where there's many of them against the concentration gradient, from low concentration to high concentration. The second protein in the lower part of the membrane is a symport pump, because it's combining both the hydrogen ion and sucrose are both moving through it at the same time, both going in the same direction. In this case, they're going into the cell. An antiporter pump, a good example of that is the sodium-potassium pump that cells use to regulate sodium and potassium concentrations. Cells want to pump sodium out of the cell. So in A, you can see 3 sodium ions have bound to the protein, and then along comes an ATP and binds one of its phosphate. And when that happens, the protein changes its shape. And now the sodium is released and pushed outside of the cell to a high concentration area of sodium. In the meantime, while this protein is open, potassium ions can bind to it. The phosphate releases, and now the protein reverts to its former shape and creates a portal to pump the potassium into the cell. So because we're moving two things through this same pump, one going in one direction, one coming back in the other, that makes this an antiport pump. Now, if we have really large molecules to move across the membrane, we're not going to be able to use a single protein or a portal, a channel protein to do this. We're going to have different mechanisms for this. And if we're taking something from inside the cell and pushing it out of the cell, we call this exocytosis. If we're taking something from the outside and we're bringing it in, we call this endocytosis. Okay? And in exocytosis, we've manufactured whatever these things are we're going to secrete. I mentioned insulin before, so let's use that as an example. Our chromosomes code for the production of the 2 polypeptide chains that make up insulin. We make them in the rough ER. In the Golgi apparatus, we put them together, making them a functional hormone, and then we package them with membrane from the Golgi. That membrane then can migrate through the cytoplasm, bind to the cell membrane, and then basically open out and release its contents outside of the cell. Once this happens, the membrane that made up the vesicle is now part of the cell membrane, so the cell just got a little bit bigger. Okay? In endocytosis, it's kind of the opposite of this. We're either going to reach around or create some kind of invagination that material can flow into, and then we're going to let the cell membrane reach around and pinch that off, and then that drifts into the cell as a vesicle. So we bring the material into the cell inside a membrane-bound vesicle, and our cell just got a little bit smaller. Now, there are 3 types of endocytosis that we want to cover. The first is phagocytosis. And in phagocytosis, what's going to happen is when something gets close to the cell that the cell senses that it wants to bring in, what it does is it starts to extend its cell membrane out and around whatever it is, and then pseudopods reach around and fuse, and then it breaks free, and now we've brought that substance into the cell in a vesicle or a vacuole. This might be a macrophage, which is part of your immune system, and this could be a bacterial particle that's somewhere in your body, and this macrophage wants to phagocytize this bacteria to bring it inside where it can destroy it and then keep you from becoming infected. And this is happening constantly in your cells, okay? So and this is typically used to bring in solid material. If we're going to bring in a lot of fluid, we're probably going to undergo pinocytosis. And in pinocytosis, instead of reaching out and around something, we're going to pull the membrane in and create an invagination, and then we'll pinch this off, and now we've got a vesicle with whatever filled this depression. And again, it can bring in solids, but most of the time this is bringing in liquids. In receptor-mediated endocytosis, there are going to be specific receptors on the surface of the cell membrane that recognize a specific substrate, something outside the cell. And when this binds to the receptor, and they're going to have a spot that wants to bind to this, they're going to trigger proteins called clathrin to bind to the inside of the membrane and start to pull it down and in. And as more and more of the substrate binds, and as more and more of the clathrin pulls it in, eventually we pull this in, wrap around, and pinch off, and have a vesicle coated with the clathrin that's holding whatever our ligand was to our receptors that were part of the membrane. And then once it's in the cell, we can fuse this to a lysosome, and we can digest it however, or do whatever we want to do. Okay? So this is a little different than typical pinocytosis in that we have to have receptors binding to the ligand to initiate this invagination. Exocytosis is again when we have something that the cell has made or it wants to get rid of. It could be waste material that it wants to get rid of. It packages in a vesicle. This vesicle migrates to the plasma membrane where the phospholipid bilayers fuse, and then it opens up and it empties out of the cell, and this vesicle now becomes part of the cell plasma membrane, and so now the cell is a little bit bigger. And again, this is important in cells that secrete products like the insulin we talked about, or any of the hormones that your body produces. This is important for getting rid of solid waste in a cell. So here are a lot of key terms for this chapter, but some of these you've seen before. Hydrophobic, hydrophilic you've seen before, and so not totally new. And then there are also some key concepts for you to focus on. Again, use these to prepare yourself for the exam.